Welcome back to the Deep Dive. Today, we're diving deep into the microscopic trenches, exploring what is, I think, arguably the most brutal arms race in biology.
Oh, absolutely. The battle between bacteria and the viruses that infect them, the phages.
And it's a conflict that's often won not by, sheer force, but by specialized molecular sabotage.
It's incredible. We're not talking about large-scale warfare here. This is precise, almost surgical attacks on the most fundamental systems in a cell.
And for decades, we've known that one of the most tempting targets for innate immunity, really across all domains of life, is the tRNA, transfer RNA.
That's right. tRNAs are they're essential. They're the molecular taxis,
Yeah.
They read the genetic code and ferry the right amino acid to build a protein.
So if you can disrupt that flow of tRNAs,
You bring protein synthesis and, by extension, viral replication to a screeching halt.
and we've seen bacteria develop tools for this, things like PRRC or Vedo-T. Even our own cells have proteins like SLF-N11 that do something similar.
Yeah.
But there's been this major gap.
A very conspicuous gap.
Right. The reigning champion of molecular immunity, CRISPR-Cas, has historically been absent from this specific kind of defense.
Exactly. CRISPR is famous for its adaptive immunity. It learns the virus's signature and then goes and destroys the viral blueprint, its DNA or its RNA.
But our source material for this deep dive, a major report published just this January 7th, 2026, details a discovery that completely flips that script.
It really does.
So our mission today is to quickly grasp this breakthrough. It's a previously uncharacterized nuclease that introduces a totally new CRISPR strategy. So who's the new player on the field?
Prepare to meet Cas12a3. It's a newly identified nuclease in the type V CRISPR family.
Okay.
And what makes it so shocking is that once it's triggered by recognizing a target RNA, say a viral transcript, it executes immunity by basically ignoring the virus.
What?
It preferentially cleaves the conserved 3-ROCCA tail of the cell's own tRNAs.
Wait, hold on. Let me make sure I'm hearing that right. So instead of destroying the invading army's plans, it shuts down the host's entire logistical supply chain.
That's it, exactly. It recognizes the invader and then focuses all its destructive power on the host cell itself.
That sounds incredibly risky,
Yeah.
but I guess brutally effective.
It's the ultimate scorched earth defense. The cell essentially commits translational suicide to make sure the phage can't replicate and escape.
Wow. Okay, let's unpack the mechanics of this. What makes Cas12a3 so distinct from its famous cousins in the CRISPR world?
It all comes down to the collateral damage. So the Cas12 family is massive. We know Cas12a, for example, is mainly a DNA-targeting enzyme.
Right.
Then you have Cas12a3. When Cas12a2 detects an RNA target, it just goes haywire. It starts indiscriminately shredding both RNA and DNA.
And indiscriminately shredding your own DNA sounds.
Yeah.
Catastrophic for a cell.
It is. When Cas12a2 starts cleaving the host's DNA, it triggers something called the SOS DNA damage response.
The SOS response.
Think of it as the cell's nuclear fire alarm. It is a complete, irrevocable system lockdown that often leads to cell death. It's the ultimate self-destruct button.
So if Cas12a2 is the nuclear option, where does Cas12a3 fit in?
Well, Cas12a3 and its close relative, Cas12a4, they found a smarter path. They're still activated by an RNA target, but the crucial difference is this. They exclusively cleave RNA. They leave the DNA completely untouched.
That is a fundamental difference. So they're RNA activated, but only RNA cleavers. How did the researchers actually confirm that Cas12a3 spares the DNA?
They ran the key test. They checked to see if it would trigger that catastrophic SOS response in E. coli, and the results were crystal clear. Unlike Cas12a2, which sends the cell into a panic, neither Cas12a3 nor Cas12a4 induced any measurable SOS response.
So the whole defense mechanism is totally different. It's not about genome stability.
Exactly. The Cas12a3 strategy is to drive anti-phage defense and growth arrest by shutting down the protein factory without damaging the factory's blueprint.
That's a powerful evolutionary leap.
It really is. And looking at the specific one they studied, BA1 Cas12a3, they noticed something else that was a bit strange. It only partially cleaved its initial viral target RNA.
So it wasn't even that good at destroying the virus's own message.
Right, which suggested its true preference, once it was activated, was for something else entirely, some other non-target RNA just floating around in the cell.
Okay, so the enzyme gets the green light from the viral RNA, and then it immediately shifts its attention to do more damage to the cell. How on earth did they pinpoint what that something else was.
They used this brilliant setup called a cell-free transcription translation system, or TXTL.
The TXTL system.
Yeah, it's a powerful tool because it lets you reconstruct the entire bacterial environment, all the enzymes, ribosomes, tRNAs, in a test tube. You get it away from the complexity of a living cell.
And why was that so important here? Why not just watch it in a live cell?
Because the cell-free system gives you exquisite timing control. So what they could do is they could activate the Cas12a3 by giving it the target RNA and then just let it sit for 4 hours.
Okay.
And after those 4 hours, they added the instructions for a green fluorescent protein, or GFP.
So that delays the whole key.
Yeah.
They gave Cas12a3 4 hours to do its collateral damage before the GFP instructions even showed up.
Precisely. If Cas12a3 was only targeting the viral script, or was just inefficient, you'd expect to see at least some GFP fluorescence.
But I'm guessing that's not what they saw.
The GFP fluorescence was completely abolished, gone, which demonstrated that Cas12a3 had already degraded RNA components that are essential for all gene expression. It had systematically shut down the entire machine.
That's a stunning piece of data. So it confirms its main job, once activated, isn't destroying the instruction manual. It's destroying the tools required to read any instruction manual.
Okay, to quickly summarize where we are in Act 1, Cas12a3 is a hyper-efficient RNA-triggered CRISPR nuclease that really breaks the mold for the type V family.
Right. It performs RNA-activated RNA cleavage,
Right.
But, and this is critical, it spares the DNA, which means it avoids that debilitating SOS response that its cousin Cas12a2 causes.
And third, it achieves its anti-phage defense by preferentially attacking essential cellular components for gene expression, which leads to this systematic, irreversible shutdown of translation and growth arrest.
Which brings us to the core of the sabotage. What specific essential RNA component is the victim of this preemptive strike?
And that takes us to Act 2. The researchers used direct RNA sequencing, which gave them this super high-resolution map of what was being cut up.
And the smoking gun pointed to?
The smoking gun pointed squarely at the victim pool, tRNAs. Specifically, they found that 27 out of 47 mapped tRNAs were significantly truncated, cut short.
27 of 47.
That is,
You're crippling the cell's protein-building capacity immediately. And the site of the cut is where the real genius of this is, right?
it is. Cas12a3 targets the most essential, universally conserved part of the molecule, the 3-RCCA tail of tRNAs. It just snips it right off, about 2 to 4 nucleotides upstream of the end.
Okay, so if the tRNA is the taxi, the CCA tail is what, the backseat where the amino acid passenger sits?
It's the charging port. It's the site that's universally required for the protein-building blug to even attach. If you cut that tail, the tRNA is instantly useless.
That's a perfect analogy. And that's why it's so potent.
Exactly. You know, other bacterial defenses often target something more specific, like the anticodon loop. And viruses can sometimes mutate or even encode their own tRNAs to get around that.
But they can't get around needing a CCA tail.
No. That sequence is fundamental for translation in almost all of life. So Cas12a3 turns this universal necessity into a potent, very difficult to circumvent shutdown switch.
So how does this molecular butcher find this specific, tiny, universally conserved tail every single time? The enzyme must have a unique structure to pull off that kind of surgery.
Absolutely. The cryo-EM structure, which let them visualize the enzyme in molecular detail, revealed the whole choreography. They saw the Cas12a3 complex bound to its guide RNA, the target RNA, and the sacrificial tRNA.
And I assume the RoVC domain is still doing the cutting?
Yes, the canonical RoVC endonuclease domain is still the molecular scissors, but the scissors are being guided by a new hand, so to speak.
The structural innovation that dictates the functional change.
Precisely. They discovered a previously undescribed structural fold, which they brilliantly named the tRNA loading domain, or TRLD. This domain is basically a molecular jig, a vice grip.
And what's the specific job of this TRLD?
The TRLD acts like a clamp. It grips the tRNA acceptor stem and, most importantly, that 3-RCCA tail. It then positions the tail perfectly between itself and the RoVC domain.
So it directs the cut.
It directs the cis phosphate, the exact spot to be broken right into the active site. It makes sure that the only thing getting cleaved is that vital tail.
A molecular jig that holds the target perfectly still for the scissors? That explains the selectivity.
And what's more, the structural analysis showed something about the tRNA's orientation. The T-arm interacts with another domain, REC2, helping to position the whole molecule, but crucially, the very end of the tail points towards a vacant cavity.
What does that suggest?
It strongly suggests Cas12a3 is cleaving-free tRNA, so the ones that are floating around uncharged and not actively being used in protein synthesis at that moment.
Which makes perfect sense. The cell relies on that pool of free tRNAs to keep making proteins. If you destroy the pool, the factory just runs out of raw material.
Exactly. And this was confirmed by looking at stress responses. I know when a cell runs out of charged tRNAs, it often triggers something called the stringent response.
Right, which is mediated by the ReLA gene. So did removing ReLA disabling that response stop the Cas12a3 defense?
No. Deleting ReLA did not impair its ability to interfere with plasmids at all.
So it's totally independent of that pathway. The shutdown is a direct result of just running out of usable tRNAs.
The mechanism is direct translational disruption, pure and simple.
Okay, Act 2 synthesized perfectly. Cas12a3 preferentially cleaves the universal 3' or CCA tail of tRNAs, causing this comprehensive cellular shutdown that isn't dependent on the traditional stress responses.
And the cryo-EM structure reveals the key, that unique tRNA loading domain, the TRLD. It's the molecular guide that ensures surgical precision.
And this strategic choice is brilliant. You're attacking a universally conserved, functionally necessary target that is just extremely difficult for viruses to get around.
It's amazing how much mileage evolution can get out of just adding one new structural domain.
Agreed. Okay, so now that we understand the structure and the target, let's talk dynamics and applications. How quickly does this thing become lethal, and how does this insight translate into tools we can actually use?
Well, what's fascinating is the activation cascade, which the researchers mapped out with multiple cryo-EM snapshots. When the target RNA binds, the enzyme undergoes these massive conformational changes.
Like what?
For example, the RAC2 domain shifts by a remarkable 28 angstroms. The cRNA3 of N shifts 22 angstroms.
Yeah.
This physical change is what opens up the machinery for the TRLD to start working.
That is a huge structural change. It's moving the protein from an inert state to an active killing machine.
But here's where it gets really interesting and incredibly efficient. After the tRNA is cleaved, the main body of the tRNA floats away, but the resulting cleaved product, specifically that 3-ACCA tail, it stays bound to the enzyme.
Wait, why would it hold on to the severed tail? That seems inefficient.
It's the opposite of inefficient. Holding onto that product actually locks Cas12a3 in its permanently activated state. It doesn't need to reset and find another viral RNA. It can just continuously capture and cleave subsequent free tRNAs without ever needing that initial trigger again.
So it's a single trigger for a continuous killing spree.
That's a good way to put it. It drives massive, non-stop collateral damage until the cell is translationally dead.
That kind of hyper-efficiency is exactly what you'd want if you were designing, say, a diagnostic tool. Which brings us to the most immediate application for Cas12a3.
Absolutely. The ability to distinguish between different collateral cleavage mechanisms allows for really powerful multiplex diagnostics.
Let's explain that term, orthogonality. So we have platforms like Cas13, which are great for detection, but they prefer linear, simple RNA sequences, right? Often just made of U's or A's.
Correct. Cas12a3's activity is completely orthogonal. It cleaves a very specific structured target, the tRNA tail. Since Cas13 ignores structures and Cas12a3 ignores those linear sequences, you can use them in the same tube at the same time.
They don't interfere with each other's signal.
They don't get in each other's way.
So how did the researchers weaponize this for diagnostics?
They created a specialized structured reporter molecule, they called it H25, that's designed to mimic the tRNA tail structure. This molecule has a fluorescent tag, and only Cas12a3 can cleave it.
Okay, so you have a custom reporter for Cas12a3.
Right, and by pairing Cas12a3 with its structured reporter, alongside Cas13a and Cas13b, each with their own preferred linear reporters, they successfully demonstrated one-pot detection of 3 distinct RNA biomarkers simultaneously.
Wow. That's a powerful leap. So you could detect, say, SARS-CoV-2, RSV, and influenza A in a single reaction.
That's what they did. Each one triggered a different fluorescent probe, all thanks to Cas12a3's unique orthogonal activity.
That's incredible.
And what's crucial is that the presence of a massive excess of human total RNA, the messy stuff you get in a clinical sample, it didn't interfere. In fact, in some cases, it actually enhanced the signal.
So this solidifies Cas12a3 as a critical, new, highly sensitive tool in the CRISPR diagnostic toolbox.
No question.
What a phenomenal discovery. I mean, Cas12a3 really represents a hyper-evolvable Cas family member. It shows us that even in these well-known enzyme classes, one small change like adding the TRLD and create a completely new immune strategy.
It's a total functional switch from broad, indiscriminate shredding to this targeted universal translational shutdown. And that's revolutionary, not just for diagnostics.
What do you mean?
Well, beyond the lab, the ability to inactivate nearly all tRNAs suggests powerful therapeutic applications, things like transcript-dependent cell arrest or selective cell elimination.
For something like highly aggressive cancer cells.
Exactly. And crucially, it does this without the dangerous side effect of causing direct DNA damage. It's a much more surgical way to shut down a specific cell population based purely on translational stress.
It's the perfect immune counterpunch then. Universally lethal to the infected cell, but only triggered by the viral presence. But if evolution created a mechanism this profoundly universal, we have to assume the arms race isn't over.
It never is, and that raises the most important question for the future, right? If the virus can't escape by just altering its own tRNA,
then what?
How long before viruses evolve an entirely new non-tRNA-based strategy to resist this kind of shutdown?
What is the next layer of immune defense waiting to be discovered that we haven't even characterized yet? We encourage you to explore the rich functional diversity of these antiviral defenses and maybe consider where this molecular arms race goes next.